Welcome back to our NCBI Bioinformatics course. In the previous introductory lecture, we discussed that what is NCBI, why it is important, how different databases of NCBI are connected with each other. Now, in this lecture, we will start our practical work. Our first database is the NCBI Gene database. Gene database is one of the most important databases for the researchers because it provides complete information about a particular gene. Now, suppose when scientists identify a gene of interest, they usually want to answer different questions, like where is the gene located in the genome, what is the function of this gene, how many transcripts does this gene produce, what protein does it encodes, is this gene associated with any disease, how can we download its DNA, its messenger RNA, or its coding sequence. So, in this lecture, we will learn how to search a gene, understand its information, how to retrieve different sequences from the gene database. So, let's start with how to search a gene in NCBI gene database. First, we will open NCBI website. On the NCBI website homepage, we can see in search bar. Here we can see there are many databases available. Today we want to study information about a specific gene. That's why we will directly go to the gene database. So select gene database. Now we need to name the gene that we want to study. BRCA1 gene in Homo sapiens. It is always important to mention the organism name because sometimes the gene name may exist in different species. So a gene name in bacteria or other organism may be same. Therefore, adding the organism may gives more accurate information. Gene, so we need to click on this. Every gene has a standardized name assigned by scientific organizations. For example, this BRCA1 is officially called BRCA1 DNA repair associated gene. Along with the full name, we can see here the gene symbol. This is the full name of the gene and this is the gene symbol. Here you can see official symbol, official gene name. Okay, next is organism Homo sapiens. This gene type, it is protein coding gene. The very start, phyletic lineage to Homo sapiens. You can see that from the very start, eukaryota. Also, next is the summary, another very important section in gene database because it provides complete information about the biological function of this gene. Here we can understand about what does this gene do, which biological process are controlled by this gene, why is this gene important. For example, BRCA1 is involved in DNA repair and a function of tumor suppressor. This information will help the researchers to understand the biological role of the gene before performing further experiments. So this is very important. Next, what we can see is the genomic context part. Here you can see genomic context. Now let's understand where this gene is located in the genome, in whole genome, genomic context, where this gene BRCA1 is located. This is the location 17q21.31. Okay, now let's understand this cytogenetic location description. This is the cytogenetic location of this gene BRCA1. What this described. Always keep it in your mind that first number indicates the chromosome number. So 17 is the chromosome number. After the chromosome number, we can see letter P or Q. Here you can see it is a Q. Sometimes it's a P or Q. The letter Q represents long arm of chromosome. While if it were a letter P, then it means the short arm of the chromosome. It means that on chromosome number 17, on the long arm side, this gene resides. This cytogenetic location actually describes the position of gene based on chromosome bending pattern that can be observed under microscope after staining. So next after Q is 21.31. This is 21 is the region on the long arm, and the 31 is the band. So this is this location. It gives the researchers a specific location, so where the gene is located. Next is gene structure. How many exons it has? Exons are total 31. Exons are actually regions that remains in the mature messenger RNA after processing while RNA splicing. So if exons are 31, then introns will be 30. So this will provide you the information about gene structure. Introns are non-coding regions that will be removed while RNA splicing. Next, very important information that genomic context will give you the neighboring genes. Here you can see this is the BRCA1 gene, and these are neighboring genes. Now, what are neighboring genes? Neighboring genes are the genes present close to our gene of interest. Here, our gene of interest is BRCA1, and these are present near our gene of interest. We can also understand here the direction of transcription. Here, you can see this gene is transcribed in this direction, while the neighboring genes, some are transcribed in this direction, some are in this. So here you can see that which is the direction, 5 to 3 prime or 3 to 5 prime, the replication direction is. The direction of transcription actually tells us the DNA stands is used as, which DNA stand can we use as a template. So during RNA synthesis, during messenger RNA synthesis. So now this is very important. Next is transcripts and products. A gene does not directly produce a protein. First, DNA is converted into messenger RNA through the process of transcription. So here you can see many transcripts of this gene. Now, a single gene can produce, you know very well that multiple transcripts through a process called alternative splicing. This means that one gene can generate different messenger RNA molecules by combining exons in different ways. These different transcripts may produce different protein isoforms. So, in the gene database, how many transcripts are produced by the gene you can also check here. What you can see here is the number of transcript. Here NM. In NCBI reference sequence transcripts, usually it indicates the accession number. If it starts with NM, it means it is the messenger RNA transcript. We are searching a gene BRCA1, so it will be the messenger RNA transcript of this gene. Now we will move toward the very important question that how can we download the DNA region, the sequence of DNA, the sequence of messenger RNA, the sequence of coding regions, regulatory regions. How can you download this? Before starting to learn how to download genomic DNA, messenger RNA, and coding sequence, we must know that what does it mean. So genomic DNA, it is a sequence which represents the complete DNA region of the gene, like all introns, all exons, all promoters, or regulatory regions. So if we are downloading genomic DNA, it means we are collecting the whole information of nucleotide sequence of that gene. If we are downloading genomic DNA whole sequence, it means we can use this information to make primers, like in primer designing, in PCR experiments, in mutation analysis. Next, moving toward messenger RNA, if we are downloading messenger RNA, what does it mean? It means that messenger RNA is actually the representation of processed transcript after removal of all introns, all non-coding regions. So, it is used, messenger RNA is used in complementary DNA synthesis in cloning experiments, and it contains only exons 5 and 3 prime UTRs because this is the mature messenger RNA. Next is coding sequence. This is very important sequence. It is a part of messenger RNA. It is the part. It actually codes for proteins. The mature messenger RNA contains, as we know very well, the mature messenger RNA contains 5 UTR, 3 UTR, and exons, while CDS is only contains the coding region, which starts with start codon and ends with stop codon. It represents only the sequence that will be translated into a final protein. Whenever you are downloading CDS DNA, coding sequence DNA, it will be used for protein prediction. It will be used for cloning purpose. It will be used in expression experiments, in protein analysis experiment. So this is the main difference between genomic DNA, messenger RNA, and coding sequence DNA. Why we need to download them? Because these all DNAs have their separate roles. We can get different kind of information from CDS, from messenger RNA, and from genomic DNA. For that, we will go to the under the genomic region section. We will go to this FASTA. Here nucleotide FASTA will be opened. Here you can see the whole sequence like CT, TC, TGG. We know that our DNA is made up of 4 nucleotides: adenine, guanine, thymine, cytosine. So here you can see the sequence of these nucleotides, whole sequence of BRCA1. You can see that. So you can directly copy the sequence here and paste it in the notepad, and also you can download it directly from here. Just we need to select the FOSTA format because FOSTA is a standard format. As you can see, I selected the FOSTA because it's a standardized format used in bioinformatics to represent any biological sequences. So now let's learn how to download messenger RNA sequence. For that, first we should know what is messenger RNA, how messenger RNA will be produced. It's according to central dogma DNA. The process of transcription occurs and messenger RNA will be produced. And that messenger RNA, we need to download the sequence of that messenger RNA. First, at what we downloaded the sequence of complete gene BRCA1. Now we want to download the coding region messenger RNA sequence. Now there are many transcripts. Suppose we just selected this one. To download the messenger RNA transcript section, first we will open this nucleotide. You want to study this transcript, you will go down and you can see the FASTA record of this messenger RNA. So you will select on this, and here you can see the sequence of messenger RNA. Here, this is the gene name present in Homo sapiens, and it's the transcript variant that you have selected, and it's the sequence of messenger RNA. Previous, again telling you, reminding you that before we have downloaded the sequence of whole gene BRCA1, now we are looking for the messenger RNA sequence. Here, this in this sequence, introns, I means non-coding regions are removed, and only exon section is here. So this exons, as you know very well, that they will remain in the mature messenger RNA after processing because they are coding regions. So this is the sequence of exons. This is the messenger RNA that you have selected. Next is how to download the CDS sequence, coding DNA sequence. Here you can see the information about CDS, like the length of CDS is how many nucleotides? 2136 nucleotides. Protein length is 711. Amino acids. Here you can get the information about this. Now, how to download this? Only the CDS region is translated into a protein. To download CDS, and you will download the direct 5 UTR FASTA sequence, 3 UTR FASTA sequence, and CDS coding region. Here you can download complete this sequence in a zip folder. Just download it and it will be downloaded in your downloadable sections. NCBI dataset zip folder. Now, in today's video, we learned about how to search a gene, how to understand the gene identity, gene function, gene symbols, gene ID, how to get information about the gene, gene summary, cytogenetic position of the gene, exons, introns, and neighboring genes, transcription direction of the gene, transcripts, and in the next lecture of this course, we will move toward NCBI database protein. We will learn about how to download the protein sequence, how to understand the protein information, and how to analyze the protein sequence, and how the gene and proteins are connected with the disease. So stay connected with the course because step by step we will learn complete bioinformatics analysis using NCBI databases. If you found this video helpful, please like, share and subscribe the Regal Biology with Khadija. And thank you for watching. I will see you in our next video.